Hello everyone, I am the educator for this question. The question says that at the sequence of steps involved in the sequencing of the genome of an organism. So we have to explain the steps that involved in the genome sequencing. So genome sequencing take place through two important methods. First method is known as expressed sequence tag. Expressed sequence tags or ESTS. The second method is known as sequential annotation. Sequence annotation. In express sequence tag, the sequence of only the coding part of the genome take place. Sequence of only coding part of genome. Coding part of genome take place. The coding part of genome means introns. Take place through express sequence tag, while sequential annotation, the whole genome goes into the process of sequencing. Whole genome is sequenced means ex- extrons plus introns are sequenced in this method. So during human genome project, the method which used is expressed sequence tag. Expressed sequence tags. So the steps that involved in this method are as follows. So the first step is total DNA is isolated from the cell. Total DNA is isolated. In this method, the cell is present in nucleus enclosing the chromatin material or DNA material. The chromatin material DNA is isolated outside the cell and then break down into several fragments, small fragments, using restriction enzyme. In second step, these restriction fragments are cloning of these using vector fragments using vector, which means the vectors are of two types, back and yak. Back means bacterial artificial chromosome and yak means yeast artificial chromosome. In this, an artificial plasmid is used which carry one of these fragments and then went into a particular cell and then multiplied to several units. They multiply into several units very rapidly. So in third step, the sequence of these fragments are taking place. Sequencing of these fragments using automated DNA sequencing means automated DNA sequencing. Automated DNA sequencing means these fragments which are multiplied using vectors plus artificial DNA libraries. Artificial DNA libraries are used. Libraries are used. In fourth step, these DNAs are arranged. Arrangement of sequencing on the basis of overlapping. On the basis of overlapping, they are placed. Different fragments are placed on the basis how they overlap on each other, and then they are sequenced using software. Using software. In the next step or the last step of DNA sequencing, the sequenced are annotated. मतलब annotated means these sequenced DNA are given numbers or are expressed like if these are the fragments first, second, third, then they will be annotated on an chromosome like the second is annotated on this chromosome and third is annotated on this chromosome and then express their functions. In this process, the whole genome sequencing is done for an organism. Hope you understand it well. Thank you.